First, I'd like to start by thanking everyone for having me here to speak today. We really like to get out and get our message out to different people. So with that, we'll take off. So basically today I'm going to talk to you about the trapping mechanics, basically collecting Aedes species versus Culex species, and we'll go from there. We're going to cover integrated mosquito management and basically why do we trap, a little background information on the different vector species that we're focused on at Harris County, the types of traps, I'll briefly mention ovid traps, mostly we'll stick with adult traps, the types of attractants, trap placements, and then basically some of my experiences trapping throughout my time. First, I'll start with a definition of integrated mosquito management from my mentor, Dr. Olson at Texas A&M University. And those of you that know him know that he's a bit of a wordsmith, so he can go on a little long. So his definition is systematic plans for effectively, efficiently, and safely using multiple tactics to manage a problem in the best interests of humans in terms of their health, economy, and or quality of life, which is what we're trying to do. We're not trying to eradicate mosquitoes. That's not feasible. It's not possible. We're trying to maintain a disease or the pest levels down to a tolerable level. These are the 5 elements of an integrated mosquito management program. You have a background knowledge, which is what we're doing now. A strong surveillance program, which is what we'll talk about because it's essentially trapping. The multi-tactic management, we're not going to get into that very much. That's beyond this talk. Continuous program evaluation and continued education. A lot of this stuff was covered very thoroughly by the previous speakers, so I won't waste too much time on this. So trapping, why do we trap? The main reason we trap is because it's the eyes of an effective program. If you don't know what's out there, you don't know what you're doing. You're just driving a truck up and down the streets and spraying blindly, the old spray and pray tactic. That's not very effective mosquito control. You're wasting chemical, you're unnecessarily exposing people to chemicals, and you may not even be hitting the species that you're trying to control. It's more accurate than other forms of surveillance. Many of you that take phone calls as a form of surveillance, you know that in lieu of nothing, it's better, but having a trap in that area is going to be better than even phone calls because different people have different tolerance levels. So, and I'll just relay this brief story. We were sampling at a tire repository. It was a scrap tire recycling facility that went defunct. So it's 10 acres of tires piled probably 2 stories high. And as you can imagine, it's popping Aedes albopictus, some Aedes aegypti, but it is just rolling these Aedes albopictus out of there. And there is a man that owns the property next door to this tire pile. And we would go out there, this is early in my career, we would go out there and speak to him. And he'd have 10 or 12 albopictus on him, biting him, taking blood meals. And he's like, yeah, they're not too bad today. And so you couple that and compare it with someone that's, in the inner loop and like the more ritzy residential areas, they get one mosquito, they're going crazy. You're getting this phone call right here, why aren't you doing anything? And so it really, until you put a trap out there, you don't know what you have out there. It'll tell you, are the mosquitoes present? It'll also tell you your species richness, which is your species composition. That can be skewed by the trap type and the attractant and even the placement. We use it in our program for disease surveillance, so we actually, we'll get into that a little later, and we use it to direct our control efforts. So, Harris County is 1,778 square miles. Harris County Public Health Environmental Services Mosquito Control Division, unlike many other county mosquito controls, we're responsible for the entire county. Even the city of Houston, the incorporated areas, everything. We're responsible for all 1,778 areas. So, how do you attack this? This is a big pizza. How do you attack a big pizza? One bite at a time. So we've divided it into 268 operational areas. And we divide these areas up into trap routes, and we trap each of these areas weekly. And so we have 134 gravids and 134 storm sewer traps that we monitor weekly, bring in and process and test and identify. So now we'll talk about the mosquito species that we're concerned with. And primarily it's Culex quinquefasciatus, which is responsible for West Nile virus and St. Louis encephalitis. Aedes aegypti, which is responsible for dengue and chikungunya. And yellow fever, even though we haven't had yellow fever, I don't know when the last yellow fever was, like around the turn of the 1900s, like 1908 or somewhere. And 80s albopictus, which is a player in dinghy and chikungunya, and a minor player in West Nile as well. So first, let's talk a little bit about these. Culex quinquefasciatus, it's a small, brown, inconspicuous mosquito. It has half-moon-shaped markings on the abdomen. It lays its eggs in egg rafts on the surface of water that's usually fouled by some kind of organic content, and the sky's the limit with the water fouling. It prefers to feed on birds. It's a non-aggressive biter, so if you send someone out, obviously, to do a landing raid in an area where you have West Nile virus, A, that's not very smart because you're exposing them to disease, and B, it's not very smart because these mosquitoes won't really respond to a landing raid. They could care less about people for the most part. So a landing raid is not a very effective for them. And they're active at crepuscular times and into the night. And this is just a picture of one of those egg rafts up close. This is a dime here that I put in for scale. It's a pretty picture. It's a top-down view. So you can see each one of these little dark dots is actually an egg that the female has laid, and they lay them in these little rafts and leave them floating on the water to emerge out. And this is what she looks like. You can barely see the half-moon shapes. I wanted to get a better picture, but I couldn't. I didn't have the photographic capabilities. But you get the idea. It's a plain brown mosquito. And don't worry, it's a lab mosquito, so it's clean. I had to let it bite me to get the picture. Next, we're going to look at Aedes aegypti, which is known as a yellow fever mosquito. It's black and silvery scale or white scale. It's got banded legs. It has a unique lyre shape on the scutum, which is the top of the thorax. The eggs are laid singly, preferably in artificial containers with those, and in close proximity with people generally because they prefer to feed on people. And they're a really sneaky biter. Though, if you've ever been out gardening or anything and you see a mosquito kind of hovering around, especially your ankles or mid-body, and it'll land briefly and you move or something and it takes off again. It could be an aegypti because they're very flighty. They won't stay there. They've been in association with people a long time. They kind of know when to zig and zag to stay out of view. And so here are the eggs of Aedes aegypti. I've shown this picture to a bunch of people, and the only comment I've ever gotten is, will you sharpen the pencil next time? Yes, I'll sharpen the pencil next time. But you can see how tiny these are. The pencil's really in there just for scale. And each one of these is an individual egg. And this is what the Aedes aegypti female looks like. You can see I don't have a picture of a live one because I can't get them to light on me. But you can see the lyre shape on the scutum here. And you can see the banded legs and the silvery scales. And then here's a little better picture trying to demonstrate that the lyre shape a little more. So it's important to keep these three in mind because I'm going to keep referring back to these three species because that's primarily what we're focused on. So Aedes albopictus is next, which is the Asian tiger mosquito. It's black and silvery scale, has banded legs, similar to Aedes aegypti, except it has one single skunk stripe running down the center. We'll look at it in a second. It lays its eggs singly on the side of containers. It's an indiscriminate feeder. If it's close to you, it's probably going to bite you. It doesn't care. Dogs, people, whatever's close to it. And they're active during the day. And the fun fact is that they were actually first noted in the U.S. in 1985 from Harris County. So you can see here some Aedes albopictus eggs. I put a dime in there so that I didn't have to hear any more pencil comments. And you can see they're really tiny. I mean, they'll fit inside the zero on a dime. I mean, they're very tiny eggs. So you can see it doesn't take a lot of real estate for these gals to lay their eggs on something. And this is what a female looks like. You can see this is the really distinct single stripe, the banded legs. This picture came out better than all the rest of them. So we have a little bit of background info. What are we going to do now? We're going to charge out there. We're going to make some trap route. We're going to throw traps out everywhere. Not so fast. We need a little more data before we run out there blindly and just start scattering traps in the wind. We need to pick the right trap. And this is just a selection of traps that I thought of off the top of my head. We have the gravid trap, which there's an example of the Ryder gravid trap over here. This is actually the Harris County gravid trap. We'll look at it a little later. It's got a slight modification. We have CDC miniature light traps. We have an underhouse trap, which Mike is probably familiar with because they were running them whenever he was there. We have a New Jersey trap, the BG Sentinel. This is an ova trap, or a little black jar for Aedes species. This, we take the trap part out of just a standard bus tub, and we can actually collect Culex eggs, which we use for our resistance testing. Omnidirectional Fay prints, this is a regular Fay prints. The commercial traps, a Wilton trap, which we'll talk a little bit about, homemade traps, red boxes, which are resting stations, even a backpack aspirator. I did that early in my career too, before I knew better. And lard can traps, sticky traps, and this, the rotator trap. I don't know if any of you guys have experience with these. These are good for telling periodicity, periodicity, when the mosquitoes are occurring at different times of the night. I can't figure this thing out for the life of me. Everyone else has no problem with it. I'll get 2 collections out of it, and then it'll stop halfway, and then I'm done. So I've never been able to get this to work right. I don't know why. So we'll just briefly mention ovitrapping. There's several advantages to ovitrapping. They're easy to set. They're relatively cheap because the attractant is generally some kind of water, because that's what the mosquitoes are after. It's good for starting colonies if you want to start a colony for some kind of resistance testing, or you want to rear them up to dose them. It'll provide you your even mix of male and female, which if you go and set traps out to try to test, you're just primarily going to get your female, so you won't be able to colonize or anything like that. And you can say positively, once you get the eggs, that the mosquitoes, you can say positively they're breeding in the area. You know for a fact they're breeding in the area. Not, some guy saying, yeah, they were here 20 years ago. You know they're in the area now. The disadvantages are that you're going to have to wait to figure out what you've got. You're going to have to rear them up, unless you have an electron microscope. You're going to have to rear them up to get an ID either in 4th instar or as adults, which takes a little more time. The reason I say it's not useful for disease surveillance in Texas, because you'd have to have some kind of vertical transmission for that to be really useful, and we don't really, the lacrosse subtype is really one of the only viruses where we have vertical transmission recorded. And so it's not really useful for disease surveillance. And you can get a mix of species and not know. If you set a cup in an area, you can get a mix of Aedes aegypti and Aedes albopictus. They pop off, you're like, wow, these all look different. Well, it's because you got lucky and got them both. So, and these are just examples of both of them. Like I said, here's our little black, I call them little black jars. Everyone always yells at me because they're not jars. We'll call them ovitraps for that reason. I try to hang them between less than 3 feet and close to the ground, but not so close that animals will drink out of them or knock them out. I try to hang them on something to stay there. And then this is just a bus tub with our gravid water in it so that we can collect Culex. So, adult trapping. There are 3, am I walking in the way of that? Sorry. There are 3 primary ways you're trying to trap these mosquitoes. You're either trying to trap a host-seeking female, one that's actually looking for blood at the time, or you're looking for one that's looking for an egg-laying site, a female that's requiring eggs, which are going to be your ovitraps, or you're looking for one seeking shelter, which are, for instance, your red boxes and your resting stations. Those are really good for, in my experience, the red boxes are really good for blood-fed Culex quinquefasciatus, Anopheles quadrimaculatus, and Culicida inornata. So, attractants, we'll just run through these real fast. I have pictures and a little more description on future slides. First is fouled water. I don't know how many of you guys have used the gravid water. It's everyone has their own recipe, and they all stink. It's amazing. Some people claim, better results than others. It really depends. Once someone finds a good recipe, they usually stick with it. We have CO2, which is known as a universal attractant, octanol, BG, lure, light, motion, and color or contrast. And we'll go through these a little more. So first is fouled water. You can see the top is a picture of our gravid attractant, which is essentially coastal Bermuda hay in water. And we let it sit for 1 to 2 weeks, and it gets ripe fast, and it smells bad. But it's great. The quinquefasciatus love it. So you can make your water from a variety of materials. Your recipe will determine what you collect generally. This down here is what we call a deciduous tea, which are oak leaves and things that we put into water for our Aedes attractants, just to kind of flavor the water a little more. It makes everyone feel better, and it does collect marginally better. Carbon dioxide is a byproduct of mammalian respiration. That's why mosquitoes have honed in on it over all of these years. It's known as a universal attractant. You can use a tank or dry ice. In Harris County, we use dry ice because, honestly, if we put one of those tanks out in the neighborhood, that would be in someone's kegerator within 3 hours. So in order to save money, dry ice is way less desirable to people. And also, the CO2 is effective up to 35 meters, I believe. So they really hone in on it from a long way and follow the plume back to the trap. And we use this on all of our traps, except for Gravid. If we put a CDC out, if we put a BG out, we use CO2 with everything. It's kind of like steroids. It's an easy cheat to increase your numbers. We have the other chemical attractants, which are octanol, which mimics chemical in mammalian breath. Also the BG lure, which mimics skin chemicals. And we use these primarily in our BG traps. You can use light. This is an experiment we're currently working on now where we're looking at different colors of light and different wavelengths to see if we can kind of differentiate between species. We have motion and color. So the color on a red box, I always wonder why it was red on the inside. And I knew that mosquitoes saw it as black, and really the only reason it's red is because it makes it easier to collect the mosquitoes that are resting inside it, and they see it as black anyways, which is genius. And then the omnidirectional phase prints. You can see with the coloration, black and white is a really big thing with Aedes species especially. Contrast, they really like contrast. And the omnidirectional or the phase prints, either one, will actually turn, and so there's a little motion included with that as well. And so that's also attractive to the mosquito species. Trap placement, it's going to make a big difference on your trap counts. I've seen people set traps in a backyard, and they're like, I'm getting, 50 mosquitoes in this trap, and someone else will go out and set the trap, and they'll get a few hundred. And it's just simply the difference of placing it in a better place. You want to put the traps where mosquitoes like to be. You don't want to put your trap in the middle of a parking lot and say, well, I didn't get any mosquitoes. Yeah, because a mosquito doesn't want to hang out in the parking lot. So, and it's very important also, so they need to be hidden. They need to have open space around them for the mosquitoes to actually get to the traps. And they need to be hidden to keep the people factor out of it. We've had the bomb squad called, well, the police, that wasn't the bomb squad. The police and fire department showed up because someone said they had a bomb in their backyard and it was one of our traps. So you really want to also make sure that your traps are always plainly marked with your organization's logo, a phone number, and what you're doing. Disease surveillance, pest surveillance, mosquito surveillance, anything. You just want to make sure that there's a name and number attached with it so that you don't get an angry call later in the week. And this isn't just an example. This is a caricature of an example, but you don't want to set your trap here. This is a good area. It's shaded. It's got a lot of plant life, so it keeps the humidity levels up, so the mosquitoes will fly around. They'll be more active to go, or more likely to go in there. And the difference between poor placement and good placement is similar to this. You can end up with a box like this in a poor area. You move that trap 100 yards over into a more shaded area, especially on the edge, and you can increase your collection immensely. So now I'll go through, we'll get to the traps. We're going to go through, these are the traps we're going to actually talk about. These are the traps that I have experience with, pretty extensive experience. And so I'll talk to you about these, and I'll go through some rough numbers to explain how well they collect or don't, how they compare to each other. And the Wilton trap, it was first described, it was originally designed as a trap for Aedes albopictus and Aedes aegypti. They found that it also collected Culex quinquefasciatus fairly well, not as well as a Gravid trap, obviously, but fairly well. The omnidirectional phase prints targets Aedes species. Your Gravid trap is going to target your Culex species like the fouled water. Your miniature light trap, if you put it above ground, it's a lottery. I mean, everything likes carbon dioxide, so it's really, you're going to get a crazy mix of species. In the storm sewers, you're going to be primarily Culex quinquefasciatus. And the BG Sentinel targets Aedes, but it does a really good job of collecting other species. So, the Wilton trap first. You can see it's just a cylinder, and you have a metal funnel that sits inside of a plastic cup with a screen bottom. And you put the funnel in the cup, you put the cup in the trap, and you let it hang. And that's what it looks like. And you let it hang out there. Like I say, we've set them out without CO2. We didn't get very good results from it as strictly a visual trap. Without CO2. With CO2, it does fairly well collecting mosquitoes. The advantages of it are it's small and compact. It hangs off of the ground, so you're not going to have ants and dogs and all kinds of stuff playing with it. It's relatively discreet, so you're not going to have people running around playing with your trap and stealing batteries and stuff. And the insects don't pass through a fan, so your insects are going to be in pretty good condition. The disadvantages are that it uses a 6-volt battery, and I don't know if you've lugged these 6 and 12-volt batteries around for a while. It can get pretty exhausting. They're heavy, they're bulky, they're kind of awkward. The collection cup is relatively small, so if you get into a situation where you're going to collect a whole bunch of mosquitoes, you'll fill the cup up. There's no rain cover, so if it rains on your specimens and you don't have it hidden appropriately, you're going to have, and then once you put it in we put all of our mosquitoes in the freezer to knock them down. And you put a bunch of wet mosquitoes in a freezer and you just have a brick of mosquitoes. It's a terrible, terrible thing. And also the plastic cups, when we knock them down in the freezer, when we take them out, we tend to get condensation on the cup, which holds on to some of your specimens and makes it a little harder to get some of the specimens out. I don't like that very much, and it requires the CO2 to perform well, in my experience. Next, we're going to look at the omnidirectional Fay prints. And that's this beast. It's actually 4 Fay Prince traps, or 2, depending on how you want to look at it, just attached together. And a normal Fay Prince is just this angle right here with this, and the collection cup sits inside it. It's got a built-in rain cover, which is good. And you can see we use these nets whenever I set out any kind of traps like that. I don't like those small plastic ones. I always use the regular CDC. I call them like a universal net because they fit on most traps, and your bugs don't stick to them as much, mosquitoes, sorry. And so you can see it uses this contrasting black panel, and that's what draws them in, the panel with the motion combined. And of course, we juice ours with CO2. So the advantages are the large contrasting panels. It's easy to see for the mosquitoes. It uses what I call the universal collection net, and it's got a built-in rain cover. The disadvantages are that it's bulky. And I don't know how many of you have ever tried to lug this out in the field. I hope you're not going far, because it's snagging on everything, and the wires are hanging and grabbing all the plants, and it's a mess. It's also highly visible to dogs and people. Everyone likes to see what that is and go up and look at it. It uses a 6-volt battery, and your insects pass through a fan. But the specimens that I've gotten out of these haven't been torn up as bad as some other traps we'll talk about in a minute. Next, we're going to talk about a gravid trap. And the original rider model is over here. You can see it's got the 2 bars, and the trap sits and actually it sits on the actual gravid bucket on the bus tub. Our model is a little different in the fact that you can see it actually has this shaped, I guess U-shaped. I don't know what color or what shape that would be. But it actually sits down in the water, and that does a couple of things for us. Since these traps sit on the ground, if an ant mound ever finds, or ants find your collections, you're going to have legs, wings, and pretty much maybe a couple of heads left. All of the good parts are going to be gone. And so this one eliminates that, because by setting this in here, it's slightly narrower than the width of the bucket, and so we have a moat around our trap, which makes it a little better and protects our collections a little more. And you can see so the stinky water goes into the bucket, the net goes on the trap tube, and it sits down in there, and everyone's happy, and it sits overnight. So the advantages of it collects your gravid females, especially Culex quincunfasciatus. They love this trap. They love the gravid water. It's a large universal collection net. The larger bucket provides your ant protection. And the disadvantages are that there's no rain protection, so you better have it underneath something, some vegetation or something to kind of shield it. The attractant is pungent. For those of you that have gravid trapped and got some on you, that's a gift that keeps on giving. And it sits on the ground, so it's susceptible to dogs. What we actually had to do at one site where a dog kept attacking our gravid nets, we just shook some of those red pepper flakes on it, and then he started leaving it alone. It was amazing. A technician is like, why don't we put red pepper flakes on it? I was like, I have never heard of that. And it worked. Huh. And on really hot days, you're going to collect other species, because other species are, and that's insects in general, they're all looking for humidity. When it's real hot and dry, they're going to move towards humidity. They don't care. And whenever they get down in that bucket, and here, let's go back. when they go down in that bucket and they get under here, it's going to suck them up in there. So you can get moths, you can get all kinds of stuff and all kinds of mosquito species in these. You just don't get very many. It's not very effective for sampling other mosquito species. So next we're going to talk about the CDC miniature light trap. And everyone is probably relatively familiar with this one. This one, I've seen them where they run off of the 6-volt batteries as well. Ours all have the D cells on board. And you basically put the net on the bottom, you flip the switch, and you're cooking. And so these can be hung either above ground, which this is not a very good place to hang it, but because it's kind of exposed, but it's just an example. Or we actually place all of ours below ground if we're sampling disease. The advantages are that it's small and compact. It's got a built-in rain shield. It's got the large universal net. The D-cell batteries are on board, so you're not lugging around a separate battery. It can be set above or below ground. The disadvantages, there's some research that says that the light may repel species. We leave the lights in ours that we place in storm sewers, and we don't have a problem. The insects pass through the fan, but again, I've never gotten specimens that are too beaten up from going through these. And the storm sewer trap sites are often in the roadways, which that's not the trap's fault, that's our placement's fault, but still the same. So next we'll look at the BG Sentinel, which is the new kid on the block as far as our trapping. And this is usually how you get it. They come in a green bag, but you pull it out of the green bag, which is, I believe, all set up over here. And so this is it all spread out. This is one of the early models, you can tell, because it has the tent poles, which I like much better than the current plastic poles. So you can see down in here, it has a little holder, a little pocket for your attractant if you want to put your BG lure, your octanol in there. You can see your tent stakes fit right in here to support the thing, because I think this is really just a modified, one of those collapsible clothes hampers is what it looks like. And then you have your collection cup, and you can see that you can see we had a little wear on this one, that there's a mesh funnel on the inside that fits on the bottom side of this collection cup, and then the collection bag fits over that. And so it's important whenever you're setting this up to make sure that your collection bag and your funnel are not touching, otherwise you're going to have 10 or 15 mosquitoes, if any. And then that's what it looks like all assembled. You put this apron here on, and then you set the collection cup on top of that, and you're ready to go. This is what ours look like generally set up. Sometimes we'll put the batteries inside them if we're feeling creative. But generally we just run CO2. We use these half-gallon igloo coolers, and with a hole drilled in the bottom, we run a little piece of hose through it and just run it into underneath the apron, and it sucks the mosquitoes in here and blows the plume out here, and it's pretty effective. So the advantages are that it's lightweight, it's collapsible. The insects don't pass through a fan. It's got the high contrast that the AD species like. The disadvantage is that it uses a 12-volt battery, which is even heavier than the 6-volt. There's not a rain cover, so you need to be sure to place it somewhere where it's not going to get rain. Your specimens in this one, and I think it may be an artifact of the fan because that fan is moving a lot of air to push the plume and everything out. It'll desiccate your specimens. So this is not a trap where, a gravid trap, you set it out, you run it, you set it out one day, you go, oh no, I forgot to get my gravid trap. You go back the next day, or you go back 2 days later, there's probably going to be mosquitoes in there, and they'll probably be alive. This one, you're toast. It just dries them out so much because it's sucking the air past. It's the only thing I can think. And it rests on the ground. Ants love these traps. Whenever you set this, you have to scout for ants. Otherwise, you're going to get bit by ants when you pick it up, and then you're going to pick up legs and wings. And it's not very robust. They tend to wear easily. They're made out of this canvas-like material, and it doesn't hold up long term. So now we'll just do some really quick trap comparisons. This is a study that we ran a while back, and we ran it between the BG Sentinel, the CDC, the Wilton, and the omnidirectional Fay Prince. And we actually set these traps out on this tire pile that I was telling you about. This is that gentleman's house that was getting 10 to 15 a minute. He's like, oh, they're not bad. But it was 10 acres of tires. It's since been closed down, and they've removed a lot of the tires. You can see this all used to be tires, but now the tires are focused primarily here and coming back here. But that's still a good probably 1.5 acres of tires. And a lot of it caught on fire probably a decade ago now. And so a lot of the tires are melted and rusted, but these tires right through here are intact and whole. This is a little melted pile. Anyways, that's not important. So you can see our 4 trap sites. We rotated those 4 trap types through these 4 trap sites. We did it for 8 weeks. And this is basically what we got. If a species was if we collected more than 500 of them, these are all raw numbers too, by the way. If we collected more than 500 of them, I put them on the pie chart. You can see we had a salt marsh bloom during the test, which wore everyone out. So that's the majority of what we caught during that. But we caught a total of 18 species. We only caught 10 of the Aedes aegypti. They've been displaced either by the albopictus or maybe drought. I'm not sure what ran them off. But they were there whenever I first started at mosquito control, and they're not there anymore. So it could be interspecific competition, I guess. But you can see quinks were Culex quinquefasciatus, Aedes vexans, Aedes albopictus was another really highly collected species. And these are those 4 sites divided, just the collection numbers, the raw numbers from each of those sites, just showing that there's not too much variance. I don't have my statistics on this with me, but there was no statistical significance between the sites. And then if you look at the trap types, you can see that the BG collected a tremendous amount of mosquitoes compared to an above-ground CDC, the omnidirectional, and the Wilton. And so what do we get from this? We got that Aedes tenuricus have no trap preference. They don't care. If it's out there, there's probably just so many of them, they're getting sucked into every trap anyways. The BGs and omnidirectional outperformed the CDCs above ground for Aedes albopictus collection. And the Aedes aegypti is no longer present at the tire pile. That's not really relevant. We missed them. And then every trap type outperformed the CDC collecting Culex quinquefasciatus above ground. So for whatever reason, a CDC above ground is not a very efficient manner to collect Culex quinquefasciatus. And so here's the next little example. We started running a trap route of 40 BG sentinels through Harris County. And so since we have 134 and 134, out of these 40 sites, 20 had CDC storm sewers in it, and 20 had gravid traps in it. And so these are just the 40 areas. You can see the total mosquitoes that we collected of Culex quinquefasciatus across all of them, and you can see their distribution. So the storm sewer collected the vast majority, the gravid was second, and the BG sentinel was third on this. Now granted, the data here is a little skewed because I was fighting with Access trying to figure it out, and I was having a hard time pulling out all of the data in a manner that I was comfortable with presenting. So that's why there aren't really too many numbers or statistics in this. This is just kind of looking at generalized trends. And you can see here the Aedes albopictus, the BG did very well. The gravid did okay. I mean, it caught a few. And the storm sewer caught, next to nothing. And the Aedes aegypti, you can see the BG outperformed for Aedes aegypti as well. And so whenever you look at these a little deeper, I took the 20 areas that had the BG sentinel and I compared them to the gravid traps in those areas. This one's the storm sewers. And you can see 99% of the quinks came out of our storm sewers. And whenever you look at the BG sentinels that were placed in that same area, the population distribution, we got the majority of, or 48% were quinks, 20% were albos. So your albopictus numbers have peaked and your aegypti. So whenever you look at the gravid trap in the same vein, you can see 97% of the gravids, or 97% of the mosquitoes collected in the gravids were Culex quinquefasciatus. And you can see here, it narrowed it down, 26% were albopictus and 7% were Aedes aegypti from the BG trap. And so, final thoughts. I don't know how I'm doing on time. Perfect, okay. Hopefully I'm a little early. We can get out of here a little earlier. The final thoughts, the right trap doesn't guarantee that you're going to have huge collections every day. There's population fluctuations in these guys. There's trap malfunctions. There's all kinds of things that can go wrong. However, the right combination of your trap type, your attractant, your placement, and your site greatly increases your odds of having solid collections and actually collecting disease mosquitoes if that's what you're after, or if you're just trying to catch some cool mosquitoes for a reference collection. The key is knowing what you're going after and selecting the appropriate trap type. And also, it's important to keep in mind that there's a learning process with all of these. Whenever I received my first BG Sentinel, they didn't have the highest build quality. They've fixed that significantly now. I plugged it in, and it wouldn't stay plugged in, so I just heat shrink around where the connection was, and it almost caught on fire. So there's a learning experience with all this stuff. A, it's engineered. Don't try to re-engineer it, is generally the number one. And the last thing is experiment, experiment, experiment. Read up on a trap, and if you think that it's going to work for you in your area, get one, try it, see what you get. That's the best way to figure out what's going to work for you. That's what we were doing with these visual attractant traps versus the BG. That's how we decided the BG is one of the better traps. And ask a lot of questions. In this industry, we have hundreds of very knowledgeable people with hundreds of years of experience, and everyone's been where you're at whenever you're trying out a new trap. Someone has tried it out, someone has experience, and someone can help you with it.